Okay, I can give you a minute. Okay. Okay. She has some.
Do you have an after?
Yeah.
You want to go first or? Okay, that's probably good.
Okay, that's okay. Hello? Excuse me, can we get started? Can you hear me? No, can you turn this up? All right, can everybody hear me now? Yes? It's all right? Okay, before class starts, actually there are going to be two quick announcements from two different student groups, I believe. So Here.
Okay. Hi, there's this flyer circulating through the class. I just encourage you to take one. There's going to be a National Day of Action in defense of integration and affirmative action on October 21st. Basically, I'm going around to classes making announcements. The attack on affirmative action and integration that began in the UC system and the college level is now extending into all of K through 12 public education. And the integration plan in San Francisco is now under attack. There are only 8 black students out of 650 entering students going into the #1 high school in San Francisco, which is fewer black students than entered there in 1954. So we're returning to separate and unequal. There's also information on how we can defeat the ban on affirmative action by the UC regents, and we have meetings every Monday to organize for the Day of Action. So I encourage you to come out and come to the meetings. Thanks.
Hi, everybody. My name is Larry Freitas. I'm from the School of Public Health. I'm a recruiter. I'm actually a grad student, too, in public health and public policy. And I'm going to make my plug for a conference that we're putting on. And this announcement is targeted to 5th years or graduating people, although younger folk can come, too. But we're having a conference October 16th from 8:30 to 4. It's a Saturday. We're going to have a keynote speaker, a panel of alumni that graduated from the School of Public Health, and a panel of students talking about the areas of concentration in the School of Public Health. So if you're really interested in public health, which I hope you are, please come out. I left flyers over there in the corner, and you need to RSVP with the grad peer advisors. I'm one of those people. If you have any questions about applying, you can call that number 2, and we'll be able to answer your questions. Okay? Thanks. Bye.
Hi again. This week, the topic is animal experimentation, and we're going to have a debate. And the basic format will be each speaker gets 20 minutes for an introduction, then 10 to 15 minutes for a response and rebuttal, and then finally a 5-minute conclusion. Our first speaker will be Dr. Carl Nicoll. Oh, sorry. I knew that. I apologize. I just made a mistake. Our first speaker will be Dr. Ray Greek. Dr. Ray Greek is a physician who is board certified in anesthesiology and pain management. He was an instructor at 2 of the United States' most prestigious medical schools and has published in the medical literature. Dr. Greek has performed experiments on animals and research with humans. Along with his wife, who is a veterinarian, he has written a book on the scientific fallacy of attempting to extrapolate the results of animal experimentation to humans. Their book, Sacred Cows and Golden Geese, is due for publication in the summer of 2000. Dr. Greek has shown not only that animal experiments are unnecessary, but that they have resulted in both direct and indirect harm to humans. Thus, these expensive and risky animal experiments produce nothing that could not be accomplished via other methods. Our second speaker will be Dr. Carl Nicoll. And Dr. Nicoll has a PhD in physiology from Michigan State University. He is a member of the faculty of UC Berkeley, formerly in the Department of Physiology and Anatomy, but currently in the Department of Integrative Biology. Dr. Nicoll is a member of the faculty of Cancer Research Laboratory at UCB, and his teaching experience includes courses in human and comparative physiology, human reproduction, endocrinology, and neuroendocrinology. And his major research interest is in the regulation of growth by hormones and growth factors in developing systems. So I would actually like to introduce you to your first speaker, Dr. Ray Greek, and I hope you have a great lecture.
Now, will this one work?
This works better.
That works better.
I don't know if that one works very well at all.
Can you hear me? Can everyone hear me okay? So the microphone's positioned all right? Great. All right. Our topic today is animal experimentation. And with regards to animal experimentation, there are actually two issues. The first issue is a scientific issue, and that is experimentation on animals necessary for medical science to advance? A purely scientific question. The second issue is a philosophical or ethical issue, and that is, regardless of the answer to number one, do animals have rights? Now, I am a physician and a scientist. I am not a philosopher, and today I am only going to address number one. My premise is as follows. Number one, for the purpose of treating human disease, the differences between animals and humans makes the extrapolation of animal data to humans highly improbable. Number two, we do not know in advance which animal will simulate the medical condition in humans. We can only know that after studies have been done in human beings. And number three, very small differences between humans and animals lead to lethal errors when applying animal data to human beings. Well, let's start with some of the propaganda that probably you have grown up with, and certainly that I grew up with. When I was a pre-medical student and even a medical student, I was told things like this. Quote, every major medical advance of this century has depended on animal research. In 1997, two scientists, Botting and Morrison, said in Scientific American, quote, in truth, there are no basic differences between the physiology of laboratory animals and humans. And this from the Foundation for Biomedical Research, which is basically a lobbying group for the animal experimentation industry, they say, quote, there are no alternatives to animal research. Well, we're going to explore some of those comments in detail today. But first, let me agree with my opponent. Animals and human beings do have things in common. Number one, we're composed of cells. Of course, so are plants. We also have a metabolism. We use ATP. We are composed of DNA, et cetera, et cetera. No one denies that we are similar to animals. In fact, humans share up to 99% of their DNA with non-human primates. Of course, we also share 75% of our genes with nematodes. And you have to remember the human genome consists of roughly 100,000 genes. But humans are the only primates not to have the glycoprotein sialic acid on the surface of their cells. A very small difference. But this very small difference may explain why non-human primates are so immune to diseases like malaria, prostate cancer, and cholera. So the question that we're going to address today is really an ontological problem. Are animals similar enough to human beings to allow extrapolation of experimental results? Now, only by comparing results can we make such a determination. Before we really get into the science, I just want to do a brief history of science. I know this is going to be nothing new to all of you in the class, but let's just do a brief review of Science 101. Science comes from the Latin verb to know, and really it's just a formalized method of understanding. It employs hypotheses, and it uses deductive reasoning. It talks about causality. Until a causal relationship is established, we assume that the relationship is only casual. For example, seat belts in automobile accidents. Seat belts are present in every automobile accident, but they don't cause every automobile accident. Likewise, Nobel Prize winners have experimented on animals, but is that why? Was that the cause of their winning the Nobel Prize? Smoking causes cancer in humans, but does it cause cancer in animals? Science also explains why small differences between animals and humans result in discrepancies in the findings. And I think we can summarize this by saying that science, as opposed to pseudoscience, relies on repeatability, parsimony, mensuration, heuristics, and consilience. Parsimony is the scientific model that, well, what parsimony means is the scientific model that explains the phenomena the simplest way wins. Generality means that the greater the range of data covered by a scientific model, the better. And consilience, of course, means that it conforms to known data in other fields. What this means is that science is predictive. An astrologer may get your horoscope right once or twice, but an astronomer will tell you where Neptune is going to be every day for the next 1,000 years. That's the difference. Science allows predictability. Now, I will admit that animals can sometimes be found that will demonstrate a concept of physiology, biochemistry, anatomy, etc., after that concept has been known from human studies. But that's not science. That is a retrospective discovery, not a prediction. That's like knowing the outcome of a ball game and betting on it afterwards. I would just like to find one bookie that would let me do that. So if a model, like an animal model of a human disease, is reliable or good, it should have predictive value. But here we have a problem, because not all animals respond the same way to medications, or surgery, or tobacco smoke. They don't even respond the same way amongst themselves. For example, Cyclosporin A is a medication that allows people to have kidney transplants, heart transplants, etc. But in 1984, in the Annals of Internal Medicine, scientists stated that the immunosuppressive effects of cyclosporin have differed considerably between species, limiting any direct inference that may be made regarding human organ transplantation. By the way, cyclosporin was discovered in soil samples from Switzerland, and it was in vitro research that revealed the chemical had immunosuppressive properties. More recently, a medication was discovered that was initially called FK506. Now it's called tacrolimus. It actually came on the market last year. Tacrolimus cannot be given to dogs because of a specific side effect that's not seen in humans. Other drugs also have these properties of being different from one species to the next. So do surgical procedures. For example, there's an operation in neurosurgery called ECIC. I actually did anesthesia for it when I was a resident. What you do is you bypass the internal carotid artery with a portion of the external carotid artery. Now, this operation was tested and perfected on dogs and rabbits. And neurosurgeons performed thousands of these things for years. And then one day somebody said, you know, I wonder if this is doing any good. And Dr. Sidney Peerless did a huge study where he showed that more patients actually died or suffered strokes because of this operation than were saved by it. But remember, the dogs did fine with the operation. Chemotherapy. Let's talk about chemotherapy for a moment. The National Cancer Institute tested 12 anti-cancer drugs that were currently used in humans on mice. Now, these are chemotherapies that for the most part work. They may not be perfect, but they do repress the cancer. Not only did they test them on mice, but they took mice that were growing 48 different kinds of human cancer. In other words, these mice had been manipulated such that they were not growing mouse tumors, they were growing the types of tumors that you and I would suffer from. In 30 out of the 48 times these drugs were tested, they were ineffective on the mice. Now, we already knew that they worked on human beings. So this means that 63% of the time the mouse models were wrong. And that's from Science, 1997. The same thing is true with carcinogens. In the 1950s, do you know who advertised cigarettes in Life magazine? Doctors. Go pull some of the old issues. You'll see a doctor in a white coat with a pack of cigarettes explaining why it's good for your health. You know why he did that? Because they tested cigarettes on dogs and other animals, and it didn't cause lung cancer. You know what? It is still extremely difficult to get lab animals to get lung cancer from smoking. Now, considering the fact that smoking is responsible for roughly 1/3 of the deaths in the United States today, I would think that that fact alone would tell us something. Let's go back to carcinogens. Of 20 compounds known not to cause cancer in humans, 19 did cause cancer in animals. And vice versa. Of 19 compounds known to be carcinogenic in humans, only 7 caused cancer using the National Cancer Institute's standard protocol. Remember, people, your astrologer will get your horoscope right every once in a while. Finally, let's talk just a moment about AIDS. The difference between AIDS in a human being and SIV in a monkey. SIV enters the cells of the rhesus monkey by binding to the CCR5 receptor. However, in human beings, HIV has to bind to 2 receptors in order to enter the cell. Now, the reason that the monkey is different from the human in this respect is because of a single amino acid. Just one. That's not much. But think for a moment. A single amino acid is the difference between people with normal hemoglobin and those with sickle cell anemia. A single amino acid is the difference between people with cystic fibrosis and people with a normal lungs and normal pancreas. So you see, it is very small differences that negate extrapolation between species. Between 1978 and 1988, 25 drugs were shown to be effective in treating stroke in animals. Problem is, none of them worked in human beings. Failures included barbiturates and MK801. But that really should not surprise us, because an official with the Food and Drug Administration has stated publicly, quote, "Most of the animal tests we accept have never been validated." Well, that's special. She goes on, "They have evolved over the past 20 years, and the FDA is comfortable with them. They may not work, but hey, I'm comfortable with it." Well, not surprisingly, the FDA conducted a huge study between 1976 and 1985. They followed 198 new medications. You know what? 52% of them were either recalled or relabeled because of severe side effects. Now, these side effects included things like death, stroke, heart attacks, liver failure necessitating liver transplant, psychotic episodes, seizures, etc., etc. We're not talking about "it made my foot itch." A pharmacologist, Dr. Ralph Haywood, who is the director of the Huntington Research Center in the UK, stated 10 years ago, quote, "The best guess for the correlation of adverse reactions in man and animal toxicity data is somewhere between 5 and 25%. Ladies and gentlemen, I'll bet a good astrologer can do that good. So people say, they say, "Well, Dr. Greek, what are the alternatives to animal research? You say that animal experimentation is not predictive and that it's not science. What are we going to do if we don't do experiments on animals?" And I say to you that that is a fallacious question. If you want to go to the moon and you went down to your neighborhood used car lot and you said to your used car salesman, "I need to go to the moon," he would say to you, "I have a nice, clean 1974 Vega." And you would say, "Yeah, but I want to go to the moon, and Vegas don't take us to the moon, do they?" And he would say to you, "Well, what are the alternatives of driving a Vega to the moon?" Well, the fallacy is that cars did not get us to the moon in the first place, and they will never get us to the moon. The reason that we have the high standard of medical care that we have today is because of things like epidemiology, in vitro research, autopsies, post-marketing drug surveillance, pathology, genetics, human autopsies, technology, clinical research, clinical observation, and on and on and on. To ask what are the alternatives to animal experimentation is simply a fallacy. Well, why does animal experimentation continue? There's a lot of reasons. Number one, we're all resistant to change. Newton's first law of motion applies to human nature just as well as it does to planets. Number two, ego. Do you think that scientists who have published 100 papers in the medical literature about animal experiments have an ego? Number three, many of us have been taught to memorize, but we have not been taught to think critically. Number four, PhDs are far removed from patient care. I'm not. Number five, I think. Yes, five. Animal experiments allows a researcher to crank out papers much quicker and therefore get promoted much faster than does clinical research. And the next to the last reason is guilt. I've talked to many animal experimenters who tell me that, well, Dr. Greek, your arguments sound good, but if you're right, why have I killed all these thousands of monkeys? Well, that's a good question. But the real reason that animal experimentation persists is the real reason that most things in life persist, and that is money. Animal research and animal experimentation is a multi-billion-dollar business, and everybody profits. Universities, individuals, animal breeders, animal suppliers, people who make the cages and equipment, people who make the food. Pharmaceutical companies also profit. For example, in 1998, the state of California was awarded greater than $1.6 billion in research grants from the National Institutes of Health. You can figure at least 50% of that was for animal experiments, and I can talk about that later if you want. Berkeley was awarded greater than $65 million. And again, you can figure at least 50% of that was because of research performed on animals. Now, do you think the state of California is going to give away $0.8 billion just because somebody gets up and says maybe it's not good for human health? And if I have any more time, I will give it back to our moderator. So if you want to switch back to the slides that Dr. Nicoll has.
I will not need that until the second part.
Okay.
Good afternoon, and I'd like to thank Michelle for inviting me to participate in this debate. I've been doing this for a number of years, and it's good to be back again. And the story I will give you is going to be very different from that which you just heard from Dr. Greek. And let me have the first slide. Can you dim the lights, please? Can we have the lights down? Front lights. Okay. Anyway, Dr. Greek, welcome to Bear Territory. I'd like to start out with some consideration of the historical background, and I need the lights down to see the slides. So can you get these flashlights? Yes, stage lights. I think the guy's an animal rights sympathizer. Can you see that? You can't control the stage lights from up here. What a great system we have here. Who can control the stage lights? I hope you're not dogging my time for this. Well, let me talk about it while you can't see it. What this shows is a curve. No. Oh, sorry, I didn't want to do that. Do we have a pointer? Like a laser pointer. Okay. Any luck in finding the control for the lights? Who's got the pointer? Oh, maybe it's in here. Don't think so. Does somebody have a pellet gun I could use? Oh, there's a light. Well, at least you can see the laser pointer. Okay, what this figure shows is the progress in medicine over the last 2,000 years, and I can describe it to you. And this curve is derived from data published by two historians, their names Hellemans and Bunch, and maybe you'll see it eventually. And what we've done is accumulated the major advances in biomedical research over the last 2,000 years as reported by these historians up through about 1986 or 1990. And the numbers here are in hundreds, so they're up to about 700 by that time. And if you can see it, the progress was extremely slow until just a few hundred years ago, and then the rate of progress in medical science became exponential. And this rapid increase in medical knowledge at that time was based to a significant degree on increased use of the scientific method and also of using animals in research. And so, for instance, the number of major medical advances that have occurred, as shown here, about 80% have occurred in the last 200 years, and half have occurred in the last century. And of that 80% of these major significant medical advances in the last couple of hundred years, about 3/4 were based directly or indirectly on animal experimentation. Now, any luck with the controls? No. Okay. Now, what have we gained from this kind of progress? Okay, this slide shows the average lifespan in ancient Roman Greece. It's 20 years. And that's because of a very high infant mortality. And then by 1900, the average lifespan had increased to 47 years, so it was about a 27-year gain. And now, currently, the average lifespan in North America and Europe is about 76 years. So in the last century or so, we've gained a considerable increase in average lifespan relative to the earlier times. And this is one of the major gains we've experienced through the advances in medical knowledge, which were based so heavily in animal experimentation. Any luck yet?
Not yet. You're still
Okay. Maybe by next semester we'll have it figured out. To give you an idea of the problem of infant mortality. These are some data published by Hugh Smith in 1774. These are very reliable data on mortality of children in London during this period of 1762 to 71. During this period, there were 16,000 children born each year on average. 8,000 survived to age 2 years, but only about 6,000 survived to age 5. So an extremely high rate of infant mortality, childhood mortality during these times. And this was one of the things that was reduced. Infant mortality was reduced because of our advances in medical knowledge. Now, there was also a very high mortality among, hallelujah. Uh-oh. Okay, there was also very high infant mortality in women during childbirth in the 1800s, and up to 35% in major cities in North America and Europe. And they were mostly due to infections or hemorrhage. And these have largely been eliminated or improved because of sanitation and hygiene, blood transfusion, and antibiotics. Now, the reason that we have enjoyed such a large increase in our lifespan, and we have reduced infant mortality and also maternal mortality, is because we have improved nutrition, our knowledge of nutrition. We've implemented public health measures of various kinds, including sanitation and hygiene, and we've improved child care practices. And these have contributed to the reduction in infant mortality. But the major effect has been public health measures, sanitation and hygiene. How do we come about to have public health measures? Well, this slide shows some of the public health measures that are in practice through most of the developed world anyway. Our drinking water is regulated to eliminate microbes and chemicals. The air quality is regulated. We've covered open sewers. We have established sanitary dumps for refuse. We regulate insects as our vectors for diseases such as mosquitoes, ticks, fleas, and so on. And we have mass inoculation programs for infectious diseases such as diphtheria and so on. Okay, how did we come about to establish these public health measures? Well, they were based on the proof of the germ theory of disease. In the past century, the medical establishment could not accept or would not accept the idea that tiny little microbes that you could only see with the aid of a powerful microscope, that these little organisms could possibly cause big, healthy animals and people to get sick and die. They thought that was absurd. But the germ theory of disease was proven by two researchers, Robert Koch and Louis Pasteur. And their proof was determined by studies in chicken cholera and anthrax in sheep. So this was animal studies for animal health and then eventually for human health, because these studies proved that these diseases were caused by microbes, and they also showed that they could immunize the animals against these microbes. Okay, these are the major killers of humans in North America and Europe prior to 1940. And so there are 8 bacterial or infectious diseases, 1 viral disease, and cancer. And most of you, I hope all of you, are protected against these diseases by immunizations. When I was a child, I lost friends and relatives to some of these diseases, like tuberculosis and so forth. But this is almost unheard of in the developed world now. And the development of protection against these diseases was all heavily based on various kinds of animal research. This is a picture of a polio ward that were found. These were found all over the US and Europe prior to the development of the polio vaccine. And each one of these cylinders contains a child who has paralytic polio, so they can't breathe for themselves. And these machines, called iron lungs, breathed for all the children. And there were over 50,000 cases a year in the US during the time before the advent of the polio vaccine. And now the number is down to just a few, or a few dozen at most. So this vaccination was a great lifesaver for the population, particularly children. Okay, some of the other benefits we've enjoyed from animal research. Okay, immunizations against these diseases, treatments of various kinds, ranging from antibiotics to tranquilizers and to hormones and so on. Here are some other benefits we've realized from animal research. Surgical procedures of various kinds, organ transplants, coronary bypass, even xenografts are now being tested as a possible means of providing organs. And other procedures, including anesthetics, various treatments for infertility, and something that's not appreciated, modern dental procedures and orthodontics were all developed using animal procedures. So if you've got braces or fillings or anything like that, they were tested in animals before they were approved for use in dentistry. Even contact lenses were tested in animals. Animals have benefited from research on animals too. So the information that's gained goes back to veterinary medicine, and these procedures, these are only a few, they're procedures that are also shared by animals. Okay, these are the major cause of death prior to 1940, as I showed you previously. Currently, I turn on overhead. These are the major causes of death in the population. So most of the infectious diseases have been essentially eliminated. Not entirely, but they're no longer major causes. So heart disease, cancer, stroke, lung disease, diabetes, liver disorders, these are primarily the diseases of the elderly. And since we are living a larger proportion of the population are living to ripe old ages, these are the kinds of diseases where the population is dying from, not infectious diseases. And so this is why the practice of medicine and the research emphasis has changed. The only one that's still present in this list that was present in the previous one is cancer. And AIDS, of course, is a relatively new one. Now, treating these diseases, heart disease, cancer, stroke, and so on, is very heavily dependent on surgical procedures, surgical techniques such as coronary bypass, heart transplants, and so on. And I think I got ahead of myself a bit. Yeah, the era of surgery requires 4 major technological advances. Sterile procedures, and that was based on research done by a Scottish researcher whose name escapes me for a moment, but anyway, Lister, after whose the Listerine's been named, and he did a lot of work on animal experiments to show how sterile surgical procedures could be developed. Anesthesia, blood transfusions, and antibiotics. With these advances, it was possible to bring in the modern era of surgery and accomplish the miracles that are accomplished in the surgical wards nowadays. So one of the procedures that's being widely used now is organ transplantation. And in order for that procedure to be successfully developed, we had to know about matching organ and tissue types. And we had to develop drugs that suppress specific immune functions, such as cyclosporin that Dr. Greek mentioned. Now, in order to understand the function of the immune system, this required extensive research primarily on mice, because it was possible to take mice and make selective defects in their immune system. And those of you who had any physiology know that the immune system is extremely complicated. We still don't understand it fully, but we acquired an understanding of the immune system function by working with mice with selected genetic and other kinds of defects. Then this enabled organ transplantation to be done successfully. But there are some problems, especially if you cross species in transplantation, because the immune system will reject foreign tissues and organs. But that might become overcome in certain cases by using this technique. And this shows the orange display here as a group of cells from a rat tumor. And these cells are encased in this white structure is a porous membrane. And then this encapsulated group of cells has been implanted into the brain of a guinea pig. And these rat cells produce dopamine. And this is a technique that's been considered as a way of treating people with Parkinson's disease because they don't produce enough dopamine in certain parts of the brain. You could implant these rat cells encased in this protective coating that's porous, but the immune system of the host can't get to the cells, so it doesn't elicit an immune response. And the same technique is also being explored for pancreatic islet transplants. And these are the cells that produce insulin. And this shows one pig cell inside a porous membrane container, and it's been implanted into monkeys, and it functions very well in monkeys. And so this is one approach being evaluated to treat people who have diabetes, which of course is due to insulin lack. Okay, the organ transplant field is really problematic. This shows the projection of the available organs for transplant in the population and the demand. And you can see by about this time, there's a huge demand relative to the availability, and it's unfortunate that we can't encourage more people to donate their organs. But one approach of solving this problem is to use xenotransplants. And this shows two pigs which have been genetically modified so that their cells express human proteins. And so these pigs, their tissues are not as severely immunogenic as ordinary pig cells, and this is being worked on as an approach to find transplant organs. And the pig is a good animal because many are used for food, and so getting organs such as hearts and lungs and livers would be a byproduct of the food industry. But xenotransplants have problems, and they are not to be taken too lightly, but it's still an approach being investigated. Okay, this is a cover of Nature, December 1994, and it shows some ordinary mice and what's called an obese mouse. And you can see this one mouse is heavier than those two ordinary ones. And this example was chosen as the cover for Nature because through studies of these obese mice, a new hormone was found just about 4 years ago. And the hormone is called leptin, and it's produced by fat cells. We've suspected a long time that such a hormone existed, but it took studies on mice to discover it. And now we know that leptin is an important regulator of appetite and of puberty in animals and people. So some people who have chronic obesity problems are benefited by injecting leptin, and others who have delayed puberty are having their puberty turned on appropriately by giving by leptin injection. So this is a case of sort of serendipitous animal experimentation leading to the discovery of this new hormone just a few years ago. Here are some other recent developments in animal models. Transgenic mice for human sickle cell anemia are being used to test treatments, and this has been a very intractable disease because there was no animal model. All they could do was they could cure sickle cell anemia in test tubes in culture, but the treatments for humans were not very good. So this animal model shows very great promise of allowing development and testing of drugs. Immune-deficient mice that can grow human tissues, normal and cancerous, and they're being used to test drugs in living organisms. And the mice have been equipped with a human immune system by genetic engineering, and they're being used in AIDS research. And, okay, studies on rats with severed spinal cords indicate that there are ways of restoring the severed nerves and reducing the damage. And so this is a very promising line of research for people with spinal cord injury. And, okay, mice have been infected with Chagas disease, a parasitic illness, afflicts 15 to 18 million people in South America. And they were cured of this illness by antifungal drugs. And there's also transgenic models for Alzheimer's disease now that are showing good promise for treating that disorder. Okay, now this cartoon illustrates the point that I want to finally end up on. Still okay time-wise. Here we have these animals. This is how they live: to eat, survive, and reproduce. But then the human animal comes along with his big brain, and he's We wonder what it's all about. So this is our adaptive advantage. This is what distinguishes us from other animals, because our brain allows us to collect information about the universe around us, both the living and the non-living. And our brain equips us with an insatiable curiosity. We need to gain knowledge. And we use the knowledge to protect ourselves. That's our adaptive advantage. And we are not totally safe from further epidemic diseases and even possible extermination as a species. 99% of all animals that ever existed on the planet have become extinct. That's the rule. And the forces of natural selection are very effective at driving all species to extinction, and we are no exception. And the factors that operate the dangers that we face are infectious diseases, and we haven't solved them all yet, parasites such as malaria and schistosomiasis, insects, which are vectors for disease, which also damage our food crops, and of course ourselves with our overpopulation and environmental destruction. You could add a fifth here, natural catastrophe, such as the comet that's thought to have wiped out the dinosaurs. That could happen again anytime. And that would be the end of us, along with everything else probably. Okay, here are some of the new infectious diseases that have come along in recent years. AIDS, Lyme disease, hantavirus, Ebola. And some of the old diseases have become resistant to antibiotics, so they're becoming a real problem, such as tuberculosis and cholera. These microorganisms evolve very rapidly, so we always have to struggle with them. It's like a red queen. You have to keep running to stay at pace with them, because we'll never get ahead of them, because they evolve too rapidly. So we have to devise new methods all the time. And to devise these new methods, we will have to do all kinds of experimentation, including animal experimentation. If we curb our freedom to use animals or any other approach to protect ourselves, then we will go back into the dark ages as far as I'm concerned. Thank you for your attention.
Excuse me if the gentleman making the comments would please respect both of the speakers and allow them to be treated with the respect that their academic careers have given them. I would appreciate it. This is a scientific debate, not a philosophical debate. I'm not here to talk about animal rights. I'm here to talk about science. Now, having said that, can you hear me? Is the microphone okay? Can the back row hear me? Okay. And could I have my slides back? Having said that, I thought it was interesting that our previous speaker started and ended, basically, with comments about animal rights. I'm not here today to talk about abortion. I'm not here to talk about capital punishment. And I'm for darn sure not here to talk about animal rights. What I'm here to talk about is science, and I will therefore proceed. The previous speaker talked, probably 50% of his lecture was on infectious diseases, so I will try and focus on that to a degree. I'll start with this slide. This is from Dr. Mark Feinberg, who is a AIDS researcher, and he says, quote, "What good does it do you to test something like a vaccine in a monkey? You find 5 or 6 years from now that it works in the monkey, and then you test it on humans, and you realize that humans behave totally differently from monkeys, so you've wasted 5 years." That is a respected current modern-day AIDS researcher. But let's talk a little bit more about infectious diseases in general. This is from the Handbook of Animal Models of Infection. And I should just point out here, Dr. Nicoll showed you a lot of pretty slides, and that's great. But I'm a physician. And let me tell you something, folks. Those of you who are trying to get into medical school and will someday go to medical school, Don't show me slides that just have print on it. If you don't have little references like this at the bottom, I don't care what your opinion is. Because we all have opinions. And if you want me to talk to you about abortion and what my opinion is, you know, buy me a beer and I'll do it. But if you want to talk to me about science, I want to see peer-reviewed journals. I want to see scientific references. I don't want to see type on a piece of paper. This is from the Handbook of Animal Models of Infection. I don't think this is an animal rights publication. Quote, up to this very day, all infectious diseases affecting humans are far from having appropriate animal models. And even in those cases where such infections are possible, the symptoms observed in animals and the course of the disease are often very different from those encountered in humans. Same book. One must be aware of the many limitations of using laboratory animals to model infectious processes. Few animal models are exact duplicates of human disease. Same book. The ideal animal model of infection will provide data that allows the outcome of therapy in humans to be predicted. For a variety of reasons, this ideal is difficult to attain. That's not People for the Ethical Treatment of Animals. That's the Handbook of Animal Models of Infection. Dr. Nichols mentioned polio. Let's talk about the polio vaccine. In 1840, polio was described as a disease of the central nervous system based on human observation. They found that if they isolated patients from non-infected populations, the spread of the disease plummeted. And in 1908, they developed an animal model. They found that animals could be infected by swabbing the virus into the nose. Unfortunately, that's not how humans were infected. They found in 1912 that the virus was actually in the human bowel. But scientists ignored this fact for decades. And this led one scientific publication to state, quote, but more than that, he held out doggedly against methods of clinical investigation which included clinical virology, in other words, human-based research. Approaches that would eventually make possible the unraveling of the whole story. Draper had gone completely over to Flexner's views on the nasal port of entry as opposed to the bowel port of entry. The clock had been set back about 25 years in polio research. In 1910, antibodies to the virus were found in recovering patients. In 1912, antibodies were also isolated in healthy humans. And this led to the theory that a mild exposure, like a vaccine, could immunize the individual. This was a key discovery. In 1937, on the other hand, animal experiments led scientists to spray a compound called zinc sulfate and picric acid into children's noses because they thought that the nose was the portal of infection. And if it was, obviously, this would kill the virus. Well, the result was that children lost their sense of smell permanently. In 1934, Dr. Brodie, and he was not the only one, ground up spinal cords of monkeys infected with polio and made a polio vaccine, right? Animals are furry-looking humans, so this should work. He tested the vaccine on monkeys, and he found that it worked great. And when he gave it to the children, it actually caused polio. The lucky ones were paralyzed. The unlucky ones died. Dr. Colmer did the same thing, and amazingly enough, he had the same result. 1941 is when the modern-day history of polio really begins. Dr. Sabin proved that the virus is not in the nasal mucosa of humans. 1941. Remember, that was about 30 years after we knew for sure that it was in the nasal mucosa of monkeys. Animal model was wrong for 30 years. How many people died of polio because of that? Autopsies and clinical research demonstrated that the virus migrates from the mouth to the bowel to the CNS. Doesn't go through the nose. Ender won the Nobel Prize for growing polio in culture. He didn't win the Nobel Prize for growing it in monkeys. He won it for growing it in culture. But note what the American Medical Association, in agreement with Dr. Nicoll, says, quote, studies using animals also resulted in a successful culture of the poliovirus. A Nobel Prize was awarded for this work in 1954. Wrong. A Nobel Prize was awarded for the work that resulted in the virus being grown in a petri dish. But don't take my word for it. Let's ask Dr. Al Sabin, who testified under oath before Congress in 1984, and this is a quote, "The work on prevention of polio was long delayed by the erroneous conception of the nature of the human disease based on misleading experimental models of the disease in monkeys." You won't hear that from people who have a vested interest in animal experimentation. Dr. Nichols mentioned the cholera vaccine and Dr. Koch. Let's look at the cholera vaccine and Dr. Koch and the germ theory of disease. In 1884, Koch studied cholera. He took 100 mice with him from England and he went to India. Well, he found a vaccine for the mice and it also worked in guinea pigs. Guess what? It killed the human beings. The reason for this was that the guinea pigs and the mice were not sensitive to some of the contaminants that were in the vaccine. We see the same thing today. The newest vaccine to hit the market is called a rotavirus vaccine, and it uses a tetravalent rhesus-based rotavirus, and it's being used for infants. Since the first time that it came out, it has resulted in a bowel condition called intussusception in approximately 100 children. That is a life-threatening event. What about vaccine testing? This says, even with a toxoid, and I'm just going to read you the bottom line, even with a toxoid assessed in one strain of mice, the immune response in humans can vary greatly. And that's from Vaccines, 1999, the third edition. I think Dr. Nichols also talked a little bit about cancer and carcinogens. But let's talk a little bit about chemotherapy. I'm sorry, it was chemotherapy he talked about. This is a quote from a Dr. Klausner, the head of the National Cancer Institute. He says, quote, and this was regarding angiostatin and endostatin. Some of you may remember this from last year. People do not understand how very far off the clinical trials of angiostatin and endostatin are. Quote, these proteins are very difficult to make. And we are working very hard to make the human versions because the mouse versions don't work in humans. Between 1970 and 1985, the National Cancer Institute tested over 500,000 compounds on animals to see if they would be effective chemotherapeutic agents. They found 80 that looked promising. And when tested, 24 actually proved to have anti-cancer activity in humans. The problem with the 24 was that 12 of them were too toxic to be used in humans. And of the 12 that remained, they were all known chemical analogs of currently used chemotherapeutic agents. The Handbook for Lab Animal Science, this book right here, says, and this is a rough quote, it said the National Cancer Institute spent billions of dollars and wasted decades of time, and now they no longer rely on animals when testing for chemotherapeutic agents. If anybody wants the exact quote, I can show it to them. How's my time? 3 minutes, okay. I found Dr. Nichols' arguments to be rather circular. We're here to debate the data as to whether or not animals are good models of human disease. I didn't see any scientific references from Dr. Nichols. All I saw was that he assumed that animal models of human disease were responsible for vaccines. He assumed that animals were responsible for our increasing our life expectancy, etc. Well, I'm a scientist and I'm a physician. I want peer-review references. I don't want opinion. What I have shown you today have been references from things like the Cancer Treatment Report, PPO updates of cancer, et cetera, et cetera. These are scientific journals. I've quoted people like Dr. Richard Klausner and others. And I would strongly recommend that you go and check out my references and see if I've misquoted the scientists, see if the studies that I have quoted are actually in the literature. But don't take my word for it, and don't take Dr. Nicoll's word for it. Find out these things for yourselves. Dr. Nichols also said that animals have benefited from animal experiments. That's a tautology. I'm not here to discuss whether animals are good models for other animals. But I'll tell you this. My wife's a veterinarian, and she'll be very quick to point out that a Great Dane suffers from a unique types of diseases compared to a beagle or compared to a bulldog. So once again, very small differences, even within the same species, will lead to divergent results. And lastly, let me just say that I saw in Dr. Nichols' slides a lot of promises for the future. And I think that he's writing a lot of checks that he's not going to be able to cash. The animal experimentation community has said time after time, well, see, here's something that's going to result in something great. The latest thing was the Alzheimer's mouse. You know, there's four clinical or not clinical symptoms, but there are four pathologies, if you will, that distinguish a brain with Alzheimer's disease from a normal brain. Do you know how many of those pathologies the so-called Alzheimer's mouse has? 2. It has 2. The Alzheimer's mouse is no more a model for Alzheimer's disease than some mouse that they've induced a stroke in. You can find 1 or 2 similarities in that mouse just as you can the Alzheimer's mouse. Think about it. Every week on CNN or Fox or whatever your news network is, some scientist comes out and says that he's had some tremendous discovery in animals that's going to cure something in human beings. Does that pan out? How many times have you heard about a new life-saving discovery in mice, and how many times have you then had a relative, a friend, or have you heard in the news a year later that it actually worked on humans? How many times? And again, ladies and gentlemen, don't take my word for this. I have references from the scientific literature. I don't have a vested interest in the outcome of this debate, and I'm happy to discuss it with you further. Thank you.
Okay, let me first say that it seems to me that the basic premise that Dr. Greek is arguing about is that animals and humans are so different that we can't learn useful information from them for medical applications. But you know, this basic premise just flies in the face of everything we know about biology from many, many centuries, that we're all fundamentally the same, and even he has even alluded to that. And so the basic knowledge we have from areas such as genetics came initially from plant studies, and then much of it came from studies on Drosophila, the fruit fly, and mice, and then also ourselves and other animals. The basic knowledge we have about how nerve cells function came from studies on invertebrates, the giant axon of squid and the giant nerve cells in the brain of Plesia, the current California sea slug. Likewise, our basic knowledge of immunology came primarily from studies in mice. And so he also claims that animal experimentation has no predictive value, which I disagree with. But for instance, every single new batch of drug, whether it's antibiotic or otherwise, every single new vaccine and every new hormone preparation is tested in animals to make sure that it is effective before it is released to the public. And if they're effective in animals, the prediction is 100% correct that they'll be effective in humans. And if they're not effective in animals, the prediction is 100% correct, they won't be effective in humans. Another argument that he made was that it seems that we're doing these animal experimentations because we're making money. We're torturing animals for pleasure and profit at taxpayers' expense for no good reason. Now, if that's the case, if animal experimentation is such a great gravy train, consider these data. Could I have the lights down, please? These are data that I gathered, Dr. Russell and I gathered from the 4 major research universities in Northern California, and they're based on data collected for the U.S. Department of Agriculture in 1990 and 1991. And there were a total of 2,369 investigators. These are principal investigators who have research grants. And of those, 55.5% do not use any animals at all. They use alternatives exclusively, and so, and also another 22% use very few animals. So 77% are apparently not clued in on this gravy train of animal experimentation. Imagine all those stupid researchers who are missing out on getting rich. Actually, I used to be down in this category here, but I haven't done any animal experimentation for almost 5 years. It hasn't made one bit of difference in my basic salary. So that's a total spurious argument. Animal experimentation is not a gravy train. Nobody needs to do it if you don't want to. Okay, can I have the lights? This little book here is called the Merck Manual. It's also called the Merck Index. This was published in 1899, 100 years old. It contains a summary of all of the medical knowledge of that time. That book has been replaced by these: the Merck Manual and the Merck Index. And the Merck Index lists 10,330 drugs and chemicals, and it has 3,000 pages. This little book has about 200 pages. The Merck Index has 3,000 pages, and naturally, of those 10,000 drugs, you're going to find some that work in humans and don't work in some animals, and some that work well in some animals and not in humans. But if a statistically valid analysis would say how many work in humans and not animals and vice versa. But you can pick out dozens that are the sort that Dr. Greek discussed in his presentation. Now, this great expansion of knowledge, as shown by these books, I maintain was brought about 75% of the advances were based directly or indirectly on animal research. Now, what does the American Medical Association say about this issue? Can I have the lights down, please? The American Medical Association published a white paper in 1988, and they state, the American Medical Association has been an outspoken proponent of biomedical research for over 100 years, and that tradition continues today. The association believes that research involving animals is absolutely essential to maintaining and improving the health of the American people. The association is opposed to any legislation or regulation that would inappropriately limit such research and actively supports all legislative efforts to ensure the continued use of animals in research while providing for their humane treatment. The American Medical Association represents over 500,000 practicing physicians in the country, and they don't have any immediate direct stake in animal experimentation. And the American Medical Association also conducted a survey about attitudes among physicians, practicing physicians, clinicians in the country regarding animal experimentation. And you can see that virtually all of them believe that animal experimentation has contributed to medical progress. They support continued use of animals in basic research and for clinical research. They support the use of animals for drug testing and for medical education. So, Dr. Greek, his opinions are that of a very tiny minority of practicing physicians in the United States. Okay, how much time do I have left? How much time? 6 minutes. Out of 15? 7 minutes. Okay. Okay, I find it interesting that Dr. Greek chose to quote a physician about AIDS research, and it turns out that he already published that quote in the website of In Defense of Animals, which he may be representing here today. And he said, this is the quote he made in his previous presentation. Dr. Mark Feinberg, a leading AIDS researcher, stated, quote, what good does it do to test a vaccine in a monkey? You find 5 or 6 years from now that it works in a monkey, and then you test it in humans, and you realize that humans behave totally differently from monkeys, so you've wasted 5 years. Monkeys do not die of AIDS, humans do. Well, it turns out that Dr. Feinberg was at UC San Francisco. He went to National Institute of Health for a period, and then he went to Emory. And so I wrote to him, and I said, hey, did you ever say that? And here's what he said. Unfortunately, this is going to be a little small, so I'll read it. I do not recall ever having made that statement that you indicated has been attributed to me. Further, I do not agree with it. There are many instances where the use of animal models research is absolutely essential for evaluating the safety and efficiency of candidate vaccines. In addition, and so on. He goes on to stress how important animal research is. But Dr. Greek cited him and gave you the reference, didn't he? Apparently, he was incorrect there. Okay, he also cited Dr. Klausner. And let me find that. When he was here last April, he cited Dr. Klausner again, and he also published, I believe he published this quotation in the website, but at least he certainly cited this from Dr. Klausner, who's the head of the National Cancer Institute in Bethesda, Maryland. And the quote is, the history of cancer research has been a history of curing cancer in the mouse. We have cured mice of cancer for decades, and it simply did not work in humans. Okay, so I corresponded with Dr. Klausner, and here's what he has to say about that quote attributed to him. That's a little faint. Let me get you a better. That's the actual on NIH letterhead. But here's what he said, blown up. I am completely convinced as to the critical importance of animal studies to cancer research. I have been critical of the scientific limitations of some aspects of transplantation models of cancer in mice, but I believe that one of the most important and fruitful areas of research in cancer is a new ability to create genetically engineered mice that have the clear capability of providing accurate and more valid models for cancer, for human cancer. I believe that the quote you refer to by Dr. Ray Greek is completely taken out of context. In fact, it is our new ability to study mouse, in particular models of cancer, that has been identified by the National Cancer Institutes as one of the 4 extraordinary opportunities in cancer research, and one that we have made a clear and pressing priority, and so on. Okay, so it seems that Dr. Greek was misled by that particular information also. Now, oh yeah, how much time I got? 3 minutes? Okay. He also went on about polio, and I won't have time to cover this entirely, but I'd like to respond to his quotation of the statement made by Dr. Albert Sabin, and this is one that's very widely used by anti-vivisectionists, animal rights activists. Okay, Sabin did say the work on prevention of polio was long delayed by an erroneous conception of the nature of the human disease based on misleading experimental models of the disease in monkeys. The vaccine was based on a tissue culture preparation, not animal experimentation. Well, and this was written, a letter by Dr. Kaufman, published in the Winston-Salem, North Carolina Journal, 1992. And so Sabin did make that statement. And it was curious because here's how Sabin responded to that in another letter, follow-up letter in the Winston-Salem Journal. He says, Dr. Kaufman correctly quoted my 1984 testimony before Congress, but he drew wrong conclusions from it. Dr. Kaufman was also wrong when he said the polio vaccine was based on a tissue culture preparation, not animal experimentation. On the contrary, my own experience of more than 60 years in biomedical research amply demonstrated that without the use of animals and of human beings, it would have been impossible to acquire the important knowledge needed to prevent such suffering and much suffering and premature death, not only among humans, but also among animals. In my 1956 paper on polio vaccines, I stated that during the preceding 4 years, approximately 9,000 monkeys, 150 chimpanzees, and 132 humans were used thus far in studies of various characteristics of different polio virus strains. These studies were necessary to solve many problems before an oral polio virus vaccine could become a reality. Now, the reason why he would state that animal experimentation had misled them, he didn't clarify from that. And unfortunately, he died in the interim, so we couldn't figure that out. But the book that Dr. Greek cited, this one, The History of Poliomyelitis, is probably the most authoritative and thorough discourse on the disease. And from that discourse, we can get an idea of what Sabin meant when he talked about being misled by the animal experimentation. And I'll just briefly go over this. The first indication that the polio was caused by an infectious agent was determined by Landsteiner and Popper, who injected spinal cord tissue from a child who died of the disease into monkeys, and 2 of them got the disease. They later showed it was a virus, and the only assay for the presence of the virus back then and even now is to inject it into monkeys. The Flexner came along, among others, and it was assumed by polio virus investigators that the nose was the major route of infection, and they didn't consider other possibilities. So they were sort of biased about this particular case. And Flexner did an experiment where he applied material containing the virus to the nose of monkeys, and they did get infection. And so they put a lot of effort into trying to prevent the nasal route of infection. And that's what Sabin meant when he said the animal experimentation misled them. But it was a matter of their view of the information. They weren't flexible enough in considering alternative possibilities such as the gastrointestinal tract. Okay, so the Enders, Robbins, and Weller did get the Nobel Prize for developing a culture system for the virus, but they used monkey kidney tissue. And monkey kidney tissue is still used to cultivate the virus because it's the best medium for creating it. And monkeys are still used to test the safety and efficacy of vaccines that are produced today. So, Sabin was not critical of animal experimentation. He was just saying that, yes, sometimes you get led astray. You can get led astray by almost any kind of experimentation, including clinical experimentation. Dr. Greek says there is no perfect animal model for certain human diseases. That's true, but you don't have to have a perfect model to have a very useful one from which you gain useful information. So, do I have any more time? Michelle, do I have any more time? No. Okay, so I will just finish here, and that'll be enough for now.
I just have a quick announcement. Could Leon Wong please come to the front, to this area right here? If you're in this class right now, could you please come? Thank you.
Could I have my slides back, please? Well, before I get the slides back, let me thank Dr. Nichols, because most of the time I don't get to talk about my quotes so much, and I like to talk about my quotes. Dr. Mark Feinberg said this in the Atlanta Journal-Constitution approximately 1 year ago. I have the original paper, and if anybody wants the exact quote, they can see me about it after the class. And I would encourage you to ask me for the exact quote, because you know something, if I'm right, and you go pull the Atlanta Journal-Constitution, and it was a 2-page article. It was this huge article where they interviewed Feinberg. Front page, and it continued. If I'm right about that, what can you conclude? And if I'm right when I say that the LA Times, which I think you have access to at this university, quoted Dr. Klausner, and that his quote was then picked up by various scientific journals, and he now denies it, can you hear me okay? Okay. What can you conclude about that? Well, what I would suggest that you can conclude is that these guys, can you still hear me? Can you hear me now? What I would suggest that you conclude is that these guys who do have a vested interest in animal experimentation and who do profit from animal experimentation slipped up. And now they're trying to deny it. And when Dr. Nichols, who made his tenure from animal experimentation, calls up and says, hey, did you say this? Oh, gosh, Dr. Nichols, I don't remember saying that. Goodness, no. Well, next spring, Continuum Press from New York City is going to publish my book, Sacred Cows and Golden Geese. And you'll find every single thing that I have said today referenced. And you can go look these things up, ladies and gentlemen, and that's what I'm telling you to do. Just because I say something doesn't mean it's true, and just because Dr. Nichols says something doesn't mean it's true. Go look it up in the literature. And if people just get up here and show you slides without showing you a reference, I'd be a little hard-pressed to believe that. With regards to what Dr. Nichols said about some of the organizations that I'm with, there's no such organization called Physicians and Lawyers for Responsible Medicine. It doesn't exist. And if you can find it, if you can find that he's right on that and that I'm wrong, then believe everything he said and don't believe a word that I've said. But I'm on the board of a lot of different groups, and I have a lot of different groups that come and ask my opinion on stuff, and I'm telling you, that's not one of them. Also, I don't deny that animal experiments advance knowledge. That's a tautology. Of course it advances knowledge. I'm just telling you that as a physician whose wife is a veterinarian, who's taught at 2 medical schools, I was talking to somebody on the way in. I did my residency at the University of Wisconsin, which is the oldest anesthesia training program in the world. The year that we took our boards, we were 2nd highest in the country. They haven't been lower than 3rd in I don't know how many years. I've taught at 2 of the most prestigious schools in the world. And you know what? None of what I did depended on animals. None of what my colleagues did depended on animals. You know, if the Democratic Party wants to do a poll that shows Al Gore is, 50 points ahead of George Bush, amazingly enough, they can come up with the data. And if the AMA or whoever wants to do a poll about animal experiments, yeah, I'm sure they can get 99% of doctors to say whatever they want to say. But keep in mind, folks, the AMA is not a scientific organization. The AMA is a lobbying group, just like the National Tobacco Institute and others. And I think that I have pointed out numerous discrepancies between the facts and what the AMA has come out and said. And interestingly enough, I have offered to debate numerous representatives of the AMA on issues just like the one Dr. Nichols and I are debating today. You know how many times I've done it? 0. And I will make that challenge again today. If any practicing physician or non-practicing physician wishes to debate me on this issue, I'm available. I do consult with animal rights groups. My wife is an animal rightist. I'm not here today to talk about animal rights. I'm here to talk about science. If animal experimentation helps human beings, then I'll be the first one to say that it does. But based on my research with animals, based on my research with humans, and based on my practice of medicine, ladies and gentlemen, and I'm the only one of the 2 debaters today who can say that. Based on my practice of medicine, animal experiments do not help human beings. Yeah, we're a little bit like a jellyfish. But if you think that the medication that I give you is going to act the same way in the jellyfish as it does in you, you are scientifically naive. Thank you very much for your attention.
Okay, I'm just going to be brief and quote from a Dr. Bessie Borwine, who teaches at medical students at the University of Western Ontario, and she's been involved in this issue for a number of years. And here's what she stated about the issue. The anti-vivisectionist animal rights activists have been wrong about every major medical advance during the past century and a half. They have never contributed 1 iota to the alleviation of pain and suffering from disease in man or animals. They have never invented a single new technique, devised a single useful methodology, proposed a single testable hypothesis, or achieved 1 prevention or cure. Their role has been to interfere with, obstruct, and hamper the honorable and triumphant progress of modern medical research. If the abolitionists, old or new, had had their way, they would have added enormously to the suffering of the animals they profess to love and who they claim to be protecting. It is biomedical science that protects humans and animals from the cruel ravages of disease. I couldn't say it better myself. Thank you.
Okay, right now we have, sorry about all of the technical difficulties that happened during this 2-hour class. Right now we're going to start a 15-minute question and answer session. And I'm going to be holding a microphone and walking around, so if you have questions, go ahead and raise your hand, and I'll come towards you, and you can address them in the microphone so everyone can benefit from your question. And before we start that, I would just personally like to thank both Dr. Greek and Dr. Nicoll for coming and speaking to our course. And I'd just like to say that we should all be careful and respectful in how we address our questions. Okay. So you guys can pass it back.
We're going to share the microphone. We probably share very little else, but we will share the microphone.
Hello. Go ahead. Anyone?
Okay, I just have to say that I'm not in favor of either Dr. Nicoll or you. I mean, I have a very
Thank you. At least you're not prejudiced.
I have a very neutral opinion about this, but I was just wondering about your comment, Dr. Greek, when you said that Dr. Nicoll got when he rebutted you about the quotes. You said that the way they appeared in the magazine, the conclusions that you made were perfectly right. But I was just wondering, based on the fact that he actually went out and researched that, can you really blame him for that? Like if he really emailed those people or wrote to them and asked them if they were really sincere in their, whatever they really thought about it, and if he got the right answers from them, can you really blame him for that?
I don't know. You're asking me to judge someone's motives.
No, I mean, I'm just saying.
But hang on.
Okay.
I can say a little more on that. You're asking me to judge motives, and obviously that's something I can't do. What I can do is tell you that I have quoted Klausner multiple times in multiple locations with the reference, and I've also quoted Feinberg multiple times in multiple locations with the reference. Dr. Nichols never called me and asked me for, maybe the place where he found it didn't have the reference on it, so maybe he was unable to look up the quote himself. But again, he didn't call me and ask me for the reference. So if you're going to call Feinberg and Klausner, great. I think that's a wonderful thing. But if you go check the Atlanta Journal-Constitution and that quote's in there, and folks, this isn't some little ditzel quote. This is a huge article, okay? And if Klausner or Feinberg didn't say what they said, I think they'd be suing those newspapers pretty quick, because I have spread the word all over the country. Every lecture I give, I quote those guys. I haven't been sued. The Atlanta Journal-Constitution hasn't been sued. Okay, you put 2 and 2 together.
Okay, I'm sure Dr. Greek will be happy to send us the actual reference to that article in the Atlanta Constitution so we can check it out. And it's interesting because when I approached both Klausner and Feinberg, they denied having said what they were supposed to have said, but that was true of others that Dr. Greek has quoted. And he published a quote from Dr. Utijian about the utility of epidemiological studies in relation to lung cancer and cigarettes. And I wrote to Dr. Utijian, and he said, this was completely taken out of context, as did Dr. Klausner. So this is we see a pattern, I think, developing here. Okay.
But again, I'm willing to say, if my references are wrong, then you believe everything this man said today. Dr. Nichols, are you willing to say that if my references are right, that I'm right? If my references are correct, if Klausner did say it and I can prove it, if Feinberg did say it and I can prove it, what does that do to your argument? And are you willing to say that, gosh, maybe those guys have a vested interest and it just colored their ability to remember? Kind of like when that guy waved his finger at the American public and said,
Can I begin a second question, please?
I didn't do it with that woman. Let him respond. It's only fair that he respond.
Send me the exact reference so I can check it out. because apparently those guys had faulty memories.
This question is directed to Dr. Greek. As a biologist, as a doctor, you must know that a lot of the knowledge we have today about biology, biochemistry, everything about science came from studies on animals, animal experimentation. Wouldn't you need to thank this animal experimentation and studies to this knowledge that you currently hold?
Well, either you weren't here for the lecture or I did a really poor job. That's what I've been talking about for the last couple hours. I've shown references from the scientific literature and from other scientists who disagree with your premise. I disagree with you when you say, oh, animal experiments have given us all these things. I don't think they have. And I back up my opinion, and I would emphasize, I haven't had any opinions up here today. If you want to talk about opinions, we'll talk about abortion and capital punishment and animal rights. Science is objective, it's not subjective. Science reduces, it's reductionistic. So what I've talked about today is science, and I've backed up my statements with references from the scientific literature. And that's what I have tried, obviously, and failed to do, is to show that no, the great discoveries of the past did not depend upon animals, and in fact, animals slowed them down.
Well, of course, I disagree with that premise, but I'd also like to say that I think a very good argument could be made that even if animal experimentation had not been of benefit to humans, I don't think anybody could deny that it has been of benefit to animals, okay? And in fact, it's been probably, in terms of numbers of subjects benefited, it's probably been of more benefit to animals than humans, because billions and billions of animals are immunized every year and are treated for diseases around the world. And so that, to me, justifies animal experimentation by itself.
Just as a follow-up to the last question, the Atlanta Journal-Constitution, September 21, 1997. Check that out. Yes, sir. It's right there.
Okay. There were two words that popped up for me when I was listening to this, and I wanted to address them both. They're going to go to both of you. The first one is the word history, and the basis of modern science, as Dr. Nicoll pointed out, came from the basis of modern biological science, thank you for whoever just fixed that in the back, but the basis of cell, I'll just yell. The basis of the modern, the cellular theory that we're all basing upon did come from research that was done on animals. Now, that's not to address whether today animal research is right or not, but I wanted to bring up the historical context, that all the education that you learned in school and in medical school was based at a point in time on animal research. And the second word I wanted to bring up that you mentioned a lot about was the word model. A model is not an exact fit. A model is a guideline that helps us to understand a situation that we don't know. And Dr. Nicoll has been claiming that animal research can be used to model different effects, not necessarily predict them. So are you denying, or are you saying that testing on animals in no way, shape, or form helps us to construct any models whatsoever or gain any insightful information whatsoever?
Okay, there are actually three questions there. Let me see if I can do them all three. Dr. Nicoll talked a lot about Koch and about, we all know Koch's postulates, the organism has to be purifiable, it has to be transmissible, blah, blah, blah. Did you know that Koch came out and said that maybe his postulates weren't exactly correct? Have you ever heard that? Do you think that's in the medical or scientific literature? Do you think I have a reference that says that Koch said that even if you cannot transmit the disease to an animal, that maybe that doesn't really mean that the disease is not present? That's my first comment. If you want more on that, I can go into more detail. Number two, the cell theory of, or the theory of cells is what modern medicine is based on, okay? You know how cells were discovered? They were discovered in plants after they used animal tissue to look for cells and couldn't find any. And I have references on that. And finally, I would just say, you keep saying, and I get kind of this gestalt from the audience that, well, Dr. Nicoll said, and Dr. Nicoll said, and well, I'm sure Dr. Nichols is a great guy. But you know, if you're going to medical school or any type of professional school, don't get up there and tell your professors what Dr. So-and-so said, all right? Because they're going to do the same thing to you that I'm going to do, and that is I'm just going to laugh you out of the room. You know, if the Journal of the American Medical Association publishes a peer-reviewed, double-blind, randomized, prospective study and comes to conclusions, then I'm going to use those conclusions in my debate. But if Dr. Smith decides that the world is flat, well, God bless him. Maybe he's a great guy, but that doesn't mean he's correct. Now, if I didn't answer all your questions, or if you want more detail, please see me afterwards. Let me just also say, folks, you've been raised, just like I was, to think that animal experiments are the greatest thing since sliced bread. Well, I went through my entire professional education thinking the same thing, and I assure you I was not convinced to the contrary in one lecture, regardless of how good-looking the man happens to be, okay?
Okay, we have about 5 more minutes. I'm sorry I can't get to everyone's question. I'm doing the best I can. We have one right here.
Can I respond to that? Briefly.
About 5 more minutes of questions.
Okay. Dr. Greek's right that Koch did modify his views about what was required to establish the presence of an infectious agent, and there are a number of infectious agents that will not cross species barriers, so that's okay. Cells, well, they saw protozoans, animal cells, about the same time as plant cells were identified, so you know I don't think that maybe Dr. Greek doesn't appreciate protozoans being animal cells. That's enough. Okay. Go ahead.
Okay, a couple of weeks ago in this class, we learned about a new surgical technique called laparoscopic surgery, and the surgeon who spoke about it said that they had to learn how to do it using animals, and I was wondering if you would have a surgeon learn how to test this technique.
Say the name of the surgery again. I'm sorry, I didn't.
Laparoscopic surgery.
Oh, laparoscopic.
Would you have a surgeon experiment this technique on humans first before testing it on animals and learning how to do it?
Yeah, laparoscopic surgery was a non-animal, non-human derived event. Laparoscopic surgeries were first begun when I was teaching at the University of Wisconsin. We did some of the first ones in the U.S., blah, blah, blah. And our surgeons learned how to do them exactly the way that all surgeons learn how to do any operation, and that is they watch other surgeons. That's how surgery residents become real surgeons. They watch them. Anybody who, if you think that you're going to go do a surgery residency and you're going to have time to go practice on pigs, you might want to consider an alternative career.
Actually, I think the question was that the procedure was developed in animals. Wasn't that it? Wasn't that the point? And I think that was true. But after it was developed, then surgeons can learn it by working on patients. But for complex surgery, such as brain surgery, the surgeons learn to stitch anastomose, tiny little blood vessels in rat brains before they ever get anywhere near a human brain. And there's plenty of other surgical techniques that require that the surgeons learn on animals first.
And there you have it, ladies and gentlemen. You can either believe Dr. Nichols, or you can believe a man who spent half of his life in the operating room. I mean, you know, this is, if you want to talk about science, I'm very willing to talk about science and back my opinions up with the scientific literature. But if you're going to talk about life's experiences, those statements amaze me, considering that I taught surgery residents for 2 decades.
This is a follow-up question to the question that was just posed. My father is a practicing physician in San Francisco, and I spoke with him about his medical school experience, and he did mention that in his first or second year, they did perform, as part of, or probably in his third year, performed surgery on animals as practice. And I think that was what, I don't want to misquote the person that just asked the question, but I think that was what he was alluding to. Do you, as a practicing physician, have qualms